So now let's move on to the final section, new modality. So we will highlight two topics. First, I'm going to talk about the Parkinson's disease cell replacement therapy, and then Shichen will take over to talk about PAH, which is another disease discussed very often in the BioSpark monthly news. So actually, this is probably the third time I cover cell replacement therapy for Parkinson's disease. For BioSpark members, you can go back to watch the previous events we talked about. In August 2023, BlueRock first announced the 12 months data for their Parkinson's disease cell replacement therapy. Then in March 2024, it's 18 months data. And finally, this April, this year, they finally published in the Nature. So, but it's also interesting to bring another competing allogeneic cell replacement trial from Japan into this highlight because they published back-to-back in the Nature. And the Japanese study is also very interesting, have some different flavor. It's also allogenic, but it's iPSC-derived dopaminergic progenitor compared to BlueRock's is the embryonic stem cell, pluripotent cell-derived DMPC. So I've tried to present the BlueRock study from many fresh new perspective, avoiding any redundancy from the previous presentations. So first is that this is a much clearer picture in terms of study design. And they have a long history. BlueRock was founded in 2016. Then 2019, they took over by Bayer and as a wholly owned subsidiary. Then they filed their IND, cleared their IND application with FDA in 2021. So they were about several years ahead of many people. And for allogenic cell replacement therapy, they do have this very streamlined manufacturing process compared to autologous iPSC-derived because the manufacturing process is just simpler. And this study design is a phase 1 study and having two subgroups. One is cohort A, having the lower dose. Here, the dose just means the total number of cells, progenitor cells, injected to the patient's putamen, one on each side. So you will have bilateral putamen injection of those DMPC cells. Then the high dose cohort is about triple the dose, about 2.7 million cells per putamen. So overall, they started the study at the day 0 for each patient, then have 1 year about immunosuppression because it's allogenic PSC-derived cells, you have to add some immunosuppression. And BlueRock's immunosuppression is a little bit more complicated. It's run in Canada, Toronto, and in New York City. They are very well-designed immunosuppression regimen, lasted for 1 year. And during the 1 year, they're adjusting dose of each immunosuppressant and for optimal consideration because as people know, unfortunately, one of the European allogenic study, STEM-PD, eventually have one patient die due to opportunistic infection. So to managing the immunosuppression regimen is very tricky and sometimes need additional care. That's why in the United States and Canada, usually they do a very sophisticated regimen. And then BlueRock announced the 12 months data in August 2023. Then around March 2024, they got the 18 months data, which looks better than 12 months data, which enables them to get the RMAT designation, Regenerative Medicine Advanced Therapy, which is the equivalent of breakthrough therapy in the cell therapy field. That's why it's quite impressive, but unfortunately, their 24 months data look a little bit not as impressive as 18 months, suggesting once you stop the immunosuppression at 12 months, the first 6 months, you may get away with the graft survival. But sounds like that 6 months, additional 6 months without immunosuppression, at least Lorenz Studer, the co-founder of BlueRock, acknowledged in a Johns Hopkins seminar that now there is definitely some worry about graft survival for the additional 6 months. But the 18 months data is good enough to get RMAT designation and published as a Nature article. And then the primary endpoint for this phase 1 is obviously safety. So you basically focus on there's no safety event, especially those serious adverse events. So overall, it's a pretty clean phase 1 study in terms of safety. Only 2 particular SAEs reported. One is due to COVID, totally irrelevant to the drug product, the cell product. The other one is after injection, after surgery, there is an epilepsy episode reported for one patient, which can be managed for additional 2 days in the hospital, eventually resolved. So that means there's basically no adverse event related to the cell replacement therapy, which is pretty good news. Then they also do neuroimaging. BlueRock did a very good job in the neuroimaging. On the top is just an illustration about the whole process, which is similar between all the cell replacement therapy for Parkinson's disease. You're just either using embryonic stem cell or the iPSC to reprogram, and then eventually differentiation is the key. Takes several months into the dopaminergic progenitor. Then you require a very skillful neurosurgeon to apply this surgery so that using the cannula to inject those cells into both sides of the brain, into the putamen area. And then you follow up with the neuroimaging. There's two types of neuroimaging conducted on this kind of setting. One is the MRI, is just to checking the safety, because MRI has the resolution, high resolution, can detect any potential tumor growth or even abnormal tissue overgrowth, especially intracerebral hemorrhage. If anything along that line happens, you will be able to see from the MRI, both on the axial view or the sagittal view. And interestingly, in the sagittal view, you can see the tract of the cell injection. Although the injected cells cannot be distinguished, the contrast agent will wear off. You will no longer tell what cells were provided from the outside, what's the endogenous human cell. You won't be able to tell that. But even at 6 months, 12 months, you can still see some tract of the original cannula injecting those cells into putamen. So that's quite interesting in terms of MRI resolution. And the PET is a functional neuroimaging. It does not have the high resolution, but people can really register and align PET images, especially F-DOPA uptake, which is a dopamine, measuring dopamine functionality. You can see how the F-DOPA was uptake by those dopaminergic neurons and leading to the different uptake signal. You can see the color gradient. Red means high uptake, blue means low uptake. And you can see that as you compare to baseline, the 12 months and 18 months, there's a slight increase across those 12 patients in the 5 patients in the low dose cohort and the 7 patients in the high dose cohort. You do see some increase, meaning those grafted dopaminergic progenitor differentiate into dopaminergic neuron and survive very well. Even at 18 months, 6 months after removal of immunosuppression, they still survive very well. So that means it's encouraging both lack of safety issues for tumor and increased uptake of dopamine function. Then one of the secondary endpoint of this BlueRock trial is some efficacy measurement. So one of the popular efficacy measurement is the so-called UPDRS Part 3 off score, just measuring when the L-DOPA drug was taken by patients. Usually when it's working, when the movement is doing well, that's called on time, meaning the drug is effective. If you're taking a drug, the patient still struggle with movement, that's called off time. So the goal is to see a reduction of off time and an increase of on time. Of course, within the on time, there's also something called good on time versus bad on time. Bad on time means there's some levodopa-induced dyskinesia. If any dyskinesia happens in the on time period, that's the bad on time. So from FDA perspective, the Hauser diary-based good on time is very much preferred endpoint. It's called approvable endpoint. If you can see some significant increase of the good on time, FDA usually likes that endpoint. But the UPDRS is more objective, it's less biased, it's the physician, neurologist really evaluate the patients and going through multiple questions and having a composite score. So in BlueRock's case, 18 months, you can see a better efficacy compared to 12 months after surgery. And the low dose cohort is less impressive, but the high dose cohort, 2.7 million per putamen, they were able to see a reduction of almost 23 points average of the off score, which is pretty good. And even better is the average increase of 2.7 hours in the good on time. So that if they can keep that up in the future readout, is even in the phase 3 trial, that's according to FDA, that's a very approvable efficacy readout. So that's overall, the 18 months looks pretty good to designate them as a RMAT breakthrough. And then in terms of other measurement of efficacy, you can think about there's a questionnaire called PDQ-39. There's 39 items you can evaluate the quality of life. BlueRock at 18 months in the high dose cohort, looking pretty good. There's some improvement of 4.2 points. May not be like a home run, but a reduction of average of 4.2 points for the PDQ-39 is also pretty good. And then they have some fluctuation in terms of the medication because patients are still taking different dosings, different formulation of the levodopa. And in the Japanese trial, I'm going to talk about later slides, is they control the medication. They are very disciplined. They think the medication dose fluctuation is a confounding factor. Whereas in BlueRock's case, they don't really strictly control the medication dose. That means there will be some fluctuation and confounding. But you can still tell that there's something called the levodopa equivalent daily dose, meaning if on average, the patient need to take less medication, there's also a credit to the cell replacement therapy. You can see on the low dose and high dose both have some reduction at 12 months. Patient on average taking less levodopa, which is good news. But unfortunately, for the 18 months data, for the high dose group, patients on average taking more. So that means there's still mixed result, although none of them may be statistically significant. But that tells you that the anti-Parkinsonian medication during the process can be a confounding factor for the eventual interpretation and the final statistical analysis of the phase 3 data. You have to take that into consideration. That's why the Japanese trial I'm going to talk now really pay attention to make sure the medication of the anti-Parkinsonia does not fluctuate. So Kyoto University Hospital, led by a principal investigator, Jun Takahashi, it's an IIT, it's an investigator-initiated. Now they call themselves a phase 1/2, and it's only done in Japan. That's why it's a Japanese RCT registration number. And they just doing those allogeneic iPSC-derived, different from BlueRock. BlueRock is embryonic stem cell derived. So they do have in Japan, because Yamanaka was the Nobel Prize winner, so he was able to build a master stem cell bank, as a master cell bank for iPSC. What's most interesting in the Japanese iPSC cell bank is they match the HLA. So according to the population genetics, immunogenetics, they just choosing those healthy donor, having the homozygous on 8 most common locus of HLA, so that in matching about 20 to 40% of Japanese population, so that it's a good position in terms of reduction of the usage of immunosuppression. If you have a lot of HLA matching, on average, you can reduce the use of immunosuppression. That's why it's a very Japanese-oriented clinical study using HLA mapping. You can view them somewhere between allogenic and autologous. They do some improved version of allogenic. So that's the unique feature of this Japanese study. But they do have they still need 12 months initial immunosuppression using tacrolimus, and reducing the dose for additional 3 months after 12 months. So a little bit more conservative because their immunosuppression regimen is less sophisticated than BlueRock. But overall, it's an interesting study because they have those different efficacy endpoints explored compared to BlueRock. But overall has less patients. Finally, you have about 6 to 7 patients eventually being looked at the efficacy cohort. And the primary endpoint, of course, is still the adverse event and the safety. So it sounds like the safety-wise is reasonably well. There's adverse events, there's no serious adverse events, but definitely there's all kinds of different adverse events, but seem to be mild, which is well tolerated for allogenic cell replacement therapy. Then for efficacy, the secondary efficacy endpoint is slightly different from BlueRock's focus. Although I think overall it's very similar. They both also do part 3 off score. Reasonably, at least 4 patients PD02, there's 4 different patients show some kind of reduction of the off score. And this is on the left is just the reduction of off score, and on the right is the percentage. They plotted into a change from baseline in terms of percentage. Overall, 4 out of 6 patients show some kind of reduction. And these are just, kind of interesting. But in terms of the UPDRS score, and it's probably less impressive. And the on-time on-score is pretty flat, not too much. There's some patients show some increase, but overall the off-score part 3 looks probably pretty good in terms of 4 out of 6. Then they also check like BlueRock in terms of the graft survival. They also do the F-DOPA imaging, but they are using a different analytical method. They compute Ki score. I don't have time to go into detail in terms of how to analyzing the DOPA neuroimaging score. But in general, you just see either increase of the Ki score in the putamen and a decrease in the caudate. And then when you do the putamen over caudate ratio, you can see many patients show pretty good increase, meaning the graft survive well in the patient's brain. Then they also, like BlueRock, checking those other measurements like the PDQ-39, like the good on time and off time. Unfortunately, in their case, compared to BlueRock, they're less impressive. They overall don't see too much improvement, even at 24 months, suggesting because BlueRock is very consistent in manufacturing from one embryonic stem cell line and doing the differentiation. Their manufacturing is probably more consistent, whereas the Japanese initiated trial, each patient receiving a different batch from the same line from the master cell bank, that sometimes they may have some struggle in terms of the consistency of those batches. But definitely you can say with a small sample size of 6 patients, they don't have a lot of improvement with the quality of life and the good on time. Meaning if it's put into USA standard, meaning the FDA probably would be less likely to approve it. But they are working with Sumitomo, a Japanese company. They can apply for a Japanese unique called fast track in Japan. So even some new Nature reported that even there's a chance, even in 2025, the Japanese agency may give them a fast track accelerated approval because the Japanese standard compared to US standard is very different. So let's see, wait and see. Overall, 2 papers published in Nature is very good news to the whole field of cell replacement therapy for Parkinson. And also, of course, sample size is small. It just warrants larger scale study. You probably need at least 100 patient plus, and you need a sham surgery because a lot of those scores are very amenable to placebo effect. So that's why it's good news to warrant future study. BlueRock will launch their phase 3 in the second half of 2025. We don't know, but the Japanese study probably will move a little bit slower. If you can get some different regulatory flexibility, they may get some accelerated approval. So that's what I'm going to share.